So the last topic to talk about in this chapter is genetics. Genetics is a rather new part of biology. It wasn't discovered in its present form until the late 1800s, and even then, unfortunately for the discoverer, it wasn't accepted as a valid explanation for inheritance. So it turns out that because of meiosis, we can predict how and we can understand how traits get passed from parents to offspring. And we can use probability actually to analyze the passage of single gene traits from parents to offspring. Gregor Mendel, a mathematician and monk, was the first scientist to actually stumble into a correct explanation for inheritance. He published his findings in 1866, and due to a major blunder in, just in that part of the world, Austria, Germany, he just wasn't taken seriously. Part of the problem was Gregor was not from the elite aristocratic uh, social class, and as a result, he just wasn't examined closely. His findings weren't, um, or his research wasn't actually tested, so no one actually determined if they could repeat it. And it wasn't until 1899, so uh, 33, you know, years later, that the science of heredity was actually discovered again by Hugo de Vries, Carl Correns, and Eric Schermark independently around the world. Mendel had already died, so he didn't get to enjoy the popularity he should have by his genius contribution to biology. Basically, what Gregor did was he studied pea plants, and thankfully he did. If he had actually gotten his way and studied mice, he probably would have figured nothing out. He developed these three basic concepts. Number one, that pea plants inherit their traits through particles, the inheritance of particles, and they do not blend these particles through reproduction. Now, you have to understand that in 1866, biologists believe in blending inheritance, which was erroneous, just like at one time, early biologists believed in spontaneous generation, and at the turn of the 20th century, biologists believed that DNA was not the hereditary chemical. In the year 2000, lots of biologists didn't believe that CRISPR-Cas9 was a gene splicing technique for viral immunity in bacteria. And so Gregor was proposing something that really kind of flew in the face of what science believed at that time, and that's part of the reason why he wasn't taken seriously. He believed that the uh, particles of inheritance segregated or separated from each other during reproduction so that each pea plant only received one, each pea plant, sperm or egg, only received one factor per trait. So when the plant made its pollen, each pollen grain only got one factor per trait. Each ovule, which is essentially an egg, only got one factor per trait. When they fuse, then the offspring pea plant cells get two factors per trait. So he's proposing that normal pea plant cells have two genetic factors per trait, but the sperm and the egg only have one factor per trait. Lastly, he proposes that the factors for different traits sort themselves out separately, and you can get all kinds of crazy combinations of traits, in the offspring. So a lot of scientists didn't look at this seriously initially to test and see if this was accurate or even true in pea plants. So basically, if we ask the question, if a green pod pea plant is crossed with a yellow pod pea plant, what will happen in the offspring? If we cross the green pod pea plant with the yellow pod pea plant, all 834 offspring turn out to be green pod. What were the genotypes of the parents? Well, it turns out that both parents are probably pure homozygous plants. The green pod plant, it turns out, is homozygous dominant, and the yellow pod plant is recessive. The offspring are all big R, little r. They're carrying particles. They have a green particle and a yellow particle, but since green dominates yellow, and I've got a spelling error here I'll have to fix. Since the green dominates the yellow, all the offspring have green pods. If two of the green pod offspring plants are crossed together with each other, you get this interesting result. 643 offspring being green pod and 214 yellow pod. It turns out the reason that happens is because the parents are what are called heterozygous. They're both big R, little r. It turns out that when the parents make their sperms and eggs, each parent has a 50% chance of passing down the big R gene and a 50% chance of passing down the little r gene. So, for example, the chance of getting an offspring that's big R is a product of the probabilities of getting a big R gene from one plant and a big R gene from the other plant. That's 50% chance of getting the big R from the one parent and 50% chance of getting big R from the other parent plant. 50% times 50% is 25%. So 25% of the offspring are predicted to be big R. 50% are predicted to be big R, little r, because one parent could produce the big R gene and the other parent the little r gene for a 50-50 shot, or 25%, but then that could reverse. The first parent could pass down the little r gene and the second parent could pass down the big R gene. The chance of that is 50% times 50% or 25%. And those two combinations do result in the big R, little r offspring. The chance of getting a little r offspring is the same as big R offspring. It's 25%. And Mendel actually then got these results, and he was able to explain them in terms of particle inheritance. The problem was nobody took him seriously. So if we mate two green wrinkled pod pea plants and we get 675 green wrinkled pod offspring, 225 green smooth pod offspring, 225 yellow wrinkled pod offspring, and 75 yellow smooth, what happened? Well, it turns out to get this combination of traits, both parents have to be heterozygous. Big R, little r, big Y, little y, and that's because we have what is called an independent assortment of traits here. If you work out how many offspring are green, it's 675 plus 225. How many are yellow? 225 plus 75. That's a 3-to-1 ratio showing that the parents were both heterozygous for color. The same thing is true for the wrinkled smooth. And as a result, then, we can say that both parents are heterozygous for both traits. And here's a graphic that kind of demonstrates that very principle there. So the pods wrinkled versus smooth and green versus yellow. Okay, now, the rules and exceptions in inheritance patterns. It turns out, and we'll have to do a number of problems in class together, and I'll probably put up a screen lecture of just how to do some of the genetics problems, but right now I just need to explain general genetics. It turns out that independent assortment will only apply to genes on different chromosomes. If two genes are on the same chromosome, they cannot be passed down separately, except if crossing over moves them around during meiosis. As a result, independent assortment generally won't happen for genes on the same chromosome. And so traits that are controlled by genes on the same chromosome tend to be inherited in sets. It says, if you perform a genetic study on fruit flies for two traits, if you start the genetics cross out at the very beginning with flies that are pure for two different traits, like parent flies are normal-winged and red-eyed or mini-winged and vermilion-eyed, the offspring are entirely normal wing and red-eyed. That means normal wings is dominant to what is called mini wings, and red eyes is dominant to vermilion. When you cross the F1 offspring flies with one another, you get this F2 generation, and it's not a independent assortment distribution. You're getting 140 normal wing red-eyed and 151 mini wing vermilion. and very few normal wing vermilion are very few mini wing red-eyed. This indicates that the genes for eyes and wings are on the same chromosome, and in the original parent, normal wings was linked to red eyes in the one parent, and mini wings was linked to vermilion eyes in the other parent. But we'll talk more about that later on. And then there's a few last things to mention here. There are human diseases that have been intensely studied, and Tay-Sachs is one of them. Tay-Sachs is a rare recessive disease that affects individuals only in the homozygous condition. So if an offspring is little a, it'll die of Tay-Sachs between the ages of 3 and 5. Basically, Tay-Sachs kids are always born to normal parents, and the parents are carriers. They're heterozygous. They're normal and maybe even healthier than people who don't carry the gene. But if two carriers marry, there's a 25% chance that the parents will produce an offspring with Tay-Sachs, because each parent has a 50% chance of passing down the Tay-Sachs gene into their sperms or eggs. That contrasts with a disease like Down syndrome, which is not inherited at all like Tay-Sachs or cystic fibrosis. Down syndrome is caused by the acquisition of an extra chromosome due to a mistake that probably is in meiosis, but it could also be in mitosis in the embryo of the baby early in development. And they call that, the baby's then a mosaic. Some of its cells are normal and some of its cells have the Down syndrome extra chromosome. So Down syndrome is not inherited as a genetic gene mistake. It's inherited as an error in the number of chromosomes that were picked up in the cell, and it's not the parents have normal chromosome counts. What happened was meiosis or mitosis screwed up, and the chromosomes were not divided properly in the M phase of the cell cycle. So today, since Down syndrome and other birth defects are not that uncommon in babies, lots of parents get prenatal testing. Prenatal testing generally involves amniocentesis, which is where a needle is stuck into the woman's abdomen and some fluid is pulled out of the baby's amniotic sac. The fluid will have baby cells in it, and you can look at the chromosomes to see if they look normal. Mom can also have a chorionic villus test done, which means that a doctor has to go up the birth canal to take a sample of cells to see if the baby's normal. So some parents actually elect to have an abortion if the baby is not normal because of the results of the prenatal test. Now, there are some unusual inheritance patterns that are caused by sex chromosomes. Sex chromosomes are chromosomes in animals that determine the gender of the animal. Gender is a complicated hereditary trait. It's not caused by a single pair of genes. It's caused by multiple genes. And generally, it's under the control of switches that are genetic switches, which are inherited on sex chromosomes. In mammals, males are said to be the heterogametic sex and females are the homogametic sex. What that means is that males have two different sex chromosomes and females have two identical sex chromosomes. In birds, it's the opposite. Birds, the males are ZZ and the females are ZW. They use different letters to avoid confusion with like XY in mammals. It turns out the Y chromosome in humans does not carry all the genes that the X does, and that means that a human male is slightly genetically defective. A human male is not fully diploid. It's missing the second copy of the genes that are on the X chromosome, and that can be a problem in inheritance. So defective X-linked genes can result in hemophilia or Duchenne muscular dystrophy or worse. And those diseases happen generally in males and not in females. And that's because males only have to get one defective X to have the disease, whereas females have to get two defective X's. Color blindness is such a disease. In humans, 1 out of about every 12 males is colorblind, but 1 out of about 140 girls is colorblind. And And that's because of the chances of getting either just one X and having the disease being a male, or having to get two bad X's and have the disease and be a female. In fruit flies, white eyes is a sex-linked trait. And by the way, the Dr. Thomas Hunt Morgan actually discovered the sex linkage phenomenon and genes being on chromosomes through a white-eyed fly and his crosses with a red-eyed fly. My sister Anne, when she was in high school, did fruit fly studies, and I was too young to quite understand what was going on, but she actually did such amazing research at Notre Dame High School that it got published, and doctors from around the country were calling to ask about her research. She went on to get her doctorate after she went to St. Louis University for free because of a full scholarship, which was pretty amazing. And then lastly down here, some traits are determined by DNA not present in the nucleus of cells. And it turns out humans carry non-nuclear DNA, and that's mitochondrial DNA. And it turns out mitochondrial DNA is easier to trace through inheritance than nuclear DNA. And so research of mitochondrial has indicated that more than likely humans originated in East Central Africa, and that's because that region of the world has the greatest diversity of mitochondrial DNA, and that's where all the migratory bands of humans originated from that carried smaller subsets of mitochondrial DNA around the world. Chloroplasts also in plants have DNA that accounts for a few plant traits in plants, but we obviously don't have chloroplasts. Chloroplast and mitochondrial DNA is not inherited in a Mendelian pattern. Mitochondrial DNA is only inherited from the mother, not from the father. Sperm do not pass any mitochondria into the fertilized egg. And in plants, if I'm not mistaken, chloroplasts are also inherited from the ovules, not the pollen grains. So in neither of those are inheritance of traits, or is inheritance of traits a Mendelian pattern.